Hello! Our topic for today is about gene regulation. Mamilet and I, Bernadette, will be your learning buddies for this particular discussion. Our first subtopic will be about how different cell types become different from each other in a multicellular organism. And this time, we will be using the carrot as a model organism. Have you ever wondered why a single cell is capable of changing to another cell type? Well, developmental biology as a science describes cell differentiation as the process through which a young and immature cell evolves into a specialized cell, reaching its mature form and function. In short, cellular differentiation is a process where a cell changes from one cell type to another, and most commonly to a more specialized type. The process of differentiation occurs numerous times during the development of a multicellular organism. The cell capable of differentiating into any type of cell is known as totipotent. Totipotency is the genetic potential of a plant cell to produce the entire plant, as in the case of the carrot illustrated in this slide. In other words, totipotency is the cell characteristic in which the potential for forming all the cell types in the adult organism is retained. The cell types in a multicellular organism becomes different from one another because they synthesize and accumulate different sets of RNA and protein molecules. This happens even without altering or changing the sequence of DNA of the different original cell before it started to differentiate. The example illustrated in this particular slide is the carrot. Cells are obtained from a transverse cut section of the carrot and are allowed to grow in culture medium containing minerals, vitamins, nutrients, and hormones to encourage cell division and growth. As a result, the cells in culture will produce a proliferative mass of cells, which is known as callus tissue. The totipotent cells of the callus tissue give rise to meristematic tissues by repeated cell divisions, giving rise to differentiated structures of the young carrot plant. Let me share with you how we differentiate transcription regulators and cis-regulatory sequences. Transcription regulators are proteins that regulate gene expression by binding to DNA in the control region of the cell. They spot or identify specific regions in the DNA, which is usually composed of 5 to 10 nucleotide pairs in length, which are also called cis-regulatory sequences. When, where, and how much a gene is expressed is controlled primarily by the interaction of cis-regulatory DNA sequences with trans-regulatory transcription factors or TFs. Our next topic is about the homeodomain protein. The homeodomain protein, as shown in the illustration, colored in orange, refers to a highly conserved region of about 60 amino acid residues that binds DNA by a helix-turn helix motif as shown in the illustration. Most homeodomain-containing proteins usually serve as transcription factors. That is, they bind to and control the activity of other genes. Many homeodomain proteins are involved in various aspects of developmental processes in many organisms. Our next subtopic is about sequence logos. What is a sequence logo? Well, perhaps in laboratory work, you got a conserved nucleic acid or protein motif that you're just dying to understand. Fear no more, because Sequence logos are here. Sequence logos are a great way to visualize large amounts of DNA or protein sequences that share in common a conserved feature. For instance, accurate gene expression relies on the work of proteins that can discern specific DNA sequences with high specificity. We call these sequences binding sites. Transcription factors are not reading DNA sequences like humans do with computers. Rather, they are moving randomly throughout the cell and interacting with the molecules in their environment. When they happen across DNA, whether or not they bind depends on the shape of the DNA, which in turn depends on the sequence. Binding might occur only if the fit is exactly right, or sometimes if the fit is about right. We can use sequence logos to understand where the transcription factor's behavior falls along this view. How do you go about a big scary data file filled with sequence information to a beautiful sequence logo? Well, take a look at the illustration. You and I will try to walk through an example of how you use sequence logos to understand these data files. On the slide, as presented here, you are facing the DNA sequences randomly generated based on weights from sequence logo, and you have the sample shown. That sequence logo actually is coming from different multiple DNA sequences. But what do you think is a real sequence logo? A real sequence logo is a great way to visualize large amounts of DNA. It may be multiple related DNA, RNA, or protein sequences, or simply DNA sequences that have common conserved binding sites, which are aligned so that the most conserved parts create good alignments. A sequence logo is a graphical display of a multiple sequence alignment consisting of color-coded stacks of letters. These letters are representing amino acids at successive positions. And sequence logos provide a richer and more precise description of sequence similarity than consensus logo sequences and can rapidly reveal significant features of the alignment that could otherwise be difficult to perceive. But if you're going to take at the illustration, you find that there are letters that are too tall while others are too short. What do they imply? Well, the total height of a logo position depends on, first, the degree of conservation in the corresponding multiple sequence alignment column, so that very conserved alignment columns produce high logo positions. Therefore, the height of each letter in a logo position is proportional to the observed frequency of the corresponding amino acid in the alignment column. The letter of each stack is ordered from most to least frequent, so that it is possible to read the consensus sequence from the top of the stacks. The next subtopic that we're going to talk about will be transcription. In this slide, we find here the illustrations of how transcription regulator will form dimers. In figure A, we have the cis regulatory sequence in the genome, and it recognizes the portion in the genome that can be identified by the transcription regulator. And when the transcription regulator identifies this, it will bind on the specific site. where the cis regulatory sequence is found. In letters B, C, and D, you will find here the dimers and the heterodimers that are facilitated by the transcription regulators. Again, in figures B, C, and D, we find here transcription regulators are forming dimers and heterodimers.
Hi, I'm going to discuss the general process on how genes can be switched on and off by an operon. But first, let us have some key points. For our first key points, let us bear in mind that bacterial genes are often found in operons. Second, each operon contains regulatory DNA sequences which act as binding sites for regulatory proteins that promote or inhibit transcription. Next, regulatory proteins often bind to small molecules which can make the protein active or inactive by changing its ability to bind DNA. And for the last one, some operons are inducible, meaning that they can be turned on by the presence of a particular small molecules. Others are repressible, meaning that they are on by default but can be turned off by a small molecule. So, how is gene expression regulated? We tend to think of bacteria as simple, but even the simplest bacterium has a complex task when it comes to gene regulation. The bacteria in your gut or between your teeth have genomes that contain thousands of different genes. Most of these genes encode proteins, each with its own role in a process such as fuel metabolism, maintenance of cell structure, and defense against viruses. Some of these proteins are needed routinely, while others are needed only at certain circumstances. Thus, cells don't express all the genes in their genome all the time. You can think of the genome as being like a cookbook with many different recipes in it. The cell will only use the recipes that fit its current needs. So how is gene expression regulated? We have here a bacterium. I'm going to discuss here the operon. Okay, so there are various forms of gene regulation. That is mechanisms for controlling which genes get expressed and at what levels. However, a lot of gene regulation occurs at the level of transcription. Bacteria have specific regulatory molecules that control whether a particular gene will be transcribed into mRNA. Often, these molecules act by binding to DNA near the gene and helping or blocking the transcription enzyme RNA polymerase. Let's take a closer look at how genes are regulated in bacteria. In bacteria, genes are often found in operons. Related genes are often found in a cluster on the chromosome where they are transcribed from one promoter RNA polymerase binding site as a single unit. Such a cluster of genes under control of a single promoter is known as an operon. Operons are common in bacteria, but they are rare in eukaryotes such as humans. In general, an operon will contain genes that function in the same process. For instance, a well-studied operon called the lac operon contains genes that encode proteins involved in uptake and metabolism of a particular sugar, lactose. Operons allow the cell to efficiently express sets of genes whose products are needed at the same time. Okay, so we are going to look at the anatomy of an operon. Operons aren't just made up of the coding sequences of genes. Instead, they also contain regulatory DNA sequences that control transcription of the operon. Typically, these sequences are binding sites for regulatory proteins, which control how much the operon is transcribed. The promoter, or site where RNA polymerase binds, is one example of a regulatory DNA sequence.
Minutes na lang before 12.
Exciting.
Most operons have other regulatory DNA sequences in addition to the promoter. These sequences are binding sites for regulatory proteins that turn expression of the operon up or down. Some regulatory proteins are repressors that bind to pieces of DNA called operators. These sequences are binding sites for regulatory proteins that turn expression of the operon up or down. Okay? Again, they are called repressors. Some regulatory proteins are repressors that bind to pieces of DNA called operators. When bound to its operator, a repressor reduces transcription, for example, by blocking RNA polymerase from moving forward on the DNA. Some regulatory proteins are activators. But where do regulatory proteins come from? Like any other protein produced in an organism, they are encoded by genes in the bacterium's genome. The genes that encode regulatory proteins are sometimes called regulatory genes. For the regulatory genes, they can themselves be turned on or off by specific small molecules. The small molecule binds to the protein, changing its shape and altering its ability to bind DNA. For instance, an activator may only become active or able to bind DNA when it's attached to a certain small molecule. Lactose. It's what's for dinner. While that may not sound delicious to us, lactose is the main sugar in milk, and you probably don't want to eat it plain. Lactose can be an excellent meal for E. coli bacteria. However, they'll only gobble up lactose when other better sugars like glucose are unavailable. With that for context, what exactly is the lac operon? The lac operon is an operon or group of genes with a single promoter transcribed as a single mRNA. The genes in the operon encode proteins that allow the bacteria to use lactose as an energy source. So what makes the lac operon turn on? E. coli bacteria can break down lactose, but it's not their favorite fuel. If glucose is around, they would much rather use that. Glucose requires fewer steps and less energy to break than lactose. However, if lactose is the only sugar available, E. coli will go right ahead and use it as an energy source. So, let us look into the structure of the lac operon. So, the lac operon contains 3 genes. LAC-Z, LAC-Y, and LAC-A. These genes are transcribed as a single mRNA under control of one promoter. Genes in the lac operon specify proteins that help the cell utilize lactose. LAC-Z encodes an enzyme that splits lactose into monosaccharides or single unit sugars that can be fed into glycolysis. Similarly, LAC-Y encodes a membrane-embedded transporter that helps bring lactose into the cell. In addition to the three genes, the lac operon also contains a number of regulatory DNA sequences. These are regions of DNA to which particular regulatory proteins can bind, controlling transcription of the operon. So let us define what is promoter. So the promoter is the binding site for RNA polymerase, the enzyme that performs transcription. What about operator? The operator is a negative regulatory site bound by the lac repressor protein. The operator overlaps with the promoter, and when the lac repressor is bound, RNA polymerase cannot bind to the promoter and start transcription. Okay, so what is a lac repressor? So I'm going to discuss the lac repressor. The lac repressor is a protein that represses or inhibits transcription of the lac operon. It does this by binding to the operator, which partially overlaps with the promoter. When bound, the lac repressor gets in RNA polymerase's way and keep it from transcribing the operon. And when lactose is not available, the lac repressor binds tightly to the operator, preventing transcription by RNA polymerase. However, when lactose is present, the lac repressor loses its ability to bind DNA. It floats off the operator, clearing the way for RNA polymerase to transcribe the operon. This change in the lac repressor is caused by the small molecule allolactose, an isomer or rearranged version of lactose. When lactose is available, some molecules will be converted to allolactose inside the cell. Allolactose binds to the lac repressor and makes it change shape so it can no longer bind DNA. It is an example of an inducer, a small molecule that triggers expression of a gene or operon. The lac operon is considered an inducible operon because it is usually turned off or repressed, but can be turned on in the presence of the inducer, allolactose. Next, I'm going to discuss catabolite activator protein or CAP. So what is CAP? So when lactose is present, the lac repressor loses its DNA binding ability. This clears the way for RNA polymerase to bind to the promoter and transcribe the lac operon. That sounds like the end of the story, right? Well, not quite. As it turns out, RNA polymerase alone does not bind very well to the lac operon promoter. It might take a few transcripts, but it won't do much more less. It gets extra help from catabolite activator protein or CAP. The CAP binds to a region of DNA just before the lac operon promoter and helps RNA polymerase attach to the promoter, driving high levels of transcription. CAP isn't always active or able to bind to DNA. Instead, it's regulated by a small molecule called cyclic AMP or CAMP. CAMP is a hunger signal made by the E. coli bacteria when glucose levels are low. CAMP binds to CAP, changing its shape and making it able to bind DNA and promote transcription. And without the CAMP or the CAMP cannot bind DNA and is inactive. CAP is only active when glucose levels are low. Thus, the lac operon can only be transcribed at high levels when glucose is absent. This strategy ensures that bacteria only turn on the lac operon and start using lactose after they have used up all of the preferred energy source or glucose. So when does the lac operon really turn on? Okay, so it must first satisfy that glucose must be unavailable. So glucose must be unavailable. When glucose is unavailable, CAMP binds to CAP, making CAP available to bind DNA. Bound CAP helps RNA polymerase attach to the lock operon promoter. And second, lactose must be available. If lactose is available, the lock repressor will be released from the operator, and this allows RNA polymerase to move forward on the DNA and transcribe the operon.
She will not talk 11.
Let us wrap it out. Okay, so let us put it all together. Now that we've seen all the moving parts of the lock operon, let's put that we've learned together to see how the operon reacts to a variety of different conditions, the presence or the absence of glucose and lactose. Okay, so first is glucose is present and lactose is absent. So if that happens, there will be no transcription of the lock operon. That's because the lock repressor remains bound to the operator and prevents transcription by RNA polymerase. Also, CAMP levels are low because glucose levels are high, so CAMP is inactive and cannot bind DNA. Next, glucose present, lactose present. So what will happen? There will be low-level transcription of the lock operon. The lock repressor is released from the operator because the inducer allolactose is present. The CAMP levels, however, are low because glucose is present. Thus, CAMP remains inactive and cannot bind to DNA, so transcription only occurs at a low liquid level. Then, for the CAP, okay, so what will happen if the glucose is absent and lactose is also absent? Okay, so there will be no transcription. No transcription of the lock operon will occur. CAMP levels are high because glucose levels are low, so CAP is active and will be bound to the DNA. However, the lock repressor will also be bound to the operator due to the absence of the allolactose. It will act as a roadblock to RNA polymerase and prevent transcription. Next, what will happen if the glucose is absent but the lactose is also absent? So, a strong transcription of the lock operon will occur. The lock repressor is released from the operator because the inducer allolactose is present. CAMP levels are high because glucose is absent, so CAP is active and bound to the DNA. CAP helps RNA polymerase bind to the promoter, permitting high levels of transcription. So here is the summary of lock operon responses. Okay, so next we have here the enhancer. Enhancer sequences are regulatory DNA sequences that, when bound by specific proteins called transcription factors, enhance the transcription of an associated gene. Regulation of transcription is the most common form of gene control, and the activity of transcription factors allow genes to be specifically regulated during development and in different types of cells. Okay, so we have here the enhancer, the data box, and the gene coding region. Okay, so for the first step, the activator protein bind to the enhancers. And actually, the enhancer is often several kilobases away from the promoter and is brought close by DNA loop. So, DNA looping brings enhancer protein complex closer. The enhancer begins to interact with mediator proteins and general transcription factors. These protein-to-protein interactions assist in the efficient assembly of the transcription initiation complex, and RNA polymerase II binds to form the initiation complex, and this will enhance the gene expression. What about the mediator? For the mediator protein complex, it is one of the prevalent co-activators that performs the biochemical task needed to initiate transcription. It is composed of more than 20 subunits. It is similar to the size of the RNA polymerase. And it also functions as a bridge between DNA-bound transcription activators, RNA polymerase, and the general transcription factors, facilitating their assembly at the promoter. So it is said that transcription activators work synergistically. Transcription activators often exhibit transcriptional synergy, wherein several DNA-bound activator proteins work together to produce a transcription rate that is much higher than the sum of their transcription rates working alone.
Let's talk about the Drosophila even skip gene. This is a very crucial gene in the development of the Drosophila embryo. When this gene is inactivated by mutation, the embryo will die early in development because many parts of the embryo will fail to progress. A study of the Stripe 2 regulatory module has provided insights into how it reads and interprets positional information. The module contains recognition sequences for two transcription regulators, bicode and hunchback, that repress it. The relative concentrations of these four proteins determine whether the protein complexes that form at the Stripe 2 module activate transcription of the EVE gene. In this particular figure, we are shown here the distributions of the four transcription regulators across the region of a Drosophila embryo where Stripe 2 forms. It is thought that either of the two repressor proteins, when bound to the DNA, will turn off the Stripe 2 module, whereas both bicoid and hunchback must bind for this module's maximal activation. This simple regulatory scheme suffices to turn on the Stripe 2 module only in those nuclei located where the levels of both bicoid and hunchback are high, and both Kruppel and giant are absent. This is a combination that occurs in only one region of the early embryo. It is not known exactly how these four transcription regulators interact with co-activators and co-repressors to specify the final level of transcription across the stripe, but the outcome very likely relies on competition between activators and repressors that act by the mechanisms as shown in the figure. Several experiments that these four regulatory proteins are responsible for the proper expression of EVE in Stripe 2. Flies that are deficient in the two gene activators bicoid and hunchback, for example, fail to efficiently express EVE in Stripe 2. In flies deficient in either of the two gene repressors, giant and Kruppel, Stripe 2 expands and covers an abnormally broad region of the embryo. Thank you for this particular learning episode. We hope that you learned something from us.
Thank you!